or should I introduce you?
Uh, uh, you
I'll
can
do
introduce me.
it. Okay. So, good evening, everyone. Welcome to the second student talk of SAC and the first biology talk of this session. We have our speaker, Shubham Kumar of Batch 18, School of Biological Sciences. I have known him for a couple of years now, and he is one of my go to persons to talk about anything biology. Last I know he was working on Drosophila tracheal system in Ranjit's lab, and today he'll be talking about the digital canvas of biology and the invisible numbers behind it. So now I would like to invite Shubham, who will help us understand what the topic is all about.
Yes, I was working with Professor Ranjit, and my prime interest is Drosophila. But since we do a lot of imaging in our lab, and due to this pandemic, I cannot do those experiments. So Professor Ranjit asked me to understand how imaging works. So that's why I did all these kind of things in my summer. And yeah, so I'll start with the title, The Digital Canvas of Biology and the Invisible Numbers Behind It. So you definitely have already been exposed to digital images. Nowadays, you will encounter them in your everyday life as well as through your scientific career. Nevertheless, be aware that it has not always been like this. 20 years ago, almost exclusively analog devices were used to acquire images. Today, we will have a hard time coming across such an analog device. Due to the widespread use of digital images, image analysis is becoming more and more important, especially in sciences, as processing digital images is much easier than processing analog ones. So this is going to be the content of my presentation. I will try to cover all of them. If uh, uh, okay, so I will start with a question. Can we trust the human eye in general while doing these kind of and scientific analysis? So the human eye and the underlying visual perception has been optimized throughout evolution. That's something we know. Moreover, it is one of the most powerful and versatile combination of detection device and analysis units attached to it. So we have a neurons, we have brain, and they work as detection device and analysis units. However, how really accurate it really comes down to quantitative image analysis. Okay, now let's start with another question. If I ask you to comment on the intensity of the little square inside the big one, what would you answer? Anyone would like to answer? Is there a difference of intensity in the lower row or either in the upper one?
No.
Okay, anyone else? What do you think? The middle boxes is there any difference uh, uh, in the upper and the
There
lower
should
box?
be. That's why you're.
Okay, so uh, actually, if we analyze the pixel intensities uh, quantitatively, it turns out uh, that the intensity is same in all six small boxes, and uh, and why this is happening, because we tend to see a decrease in intensity in the upper row. Uh, Obviously, the interpretation of the human eye is sensible to the context. As the surrounding square is getting less gray, we are having the same impression for the other one. The reason is that visual perception is optimized when seeing differences in the scene. This has been surely an advantage in former times in order to see enemies or identify a trait. But nowadays, in particular in science, it is a dangerous feature. So basically, it is one of the way our eyes are evolved, and you have come across many this such kind of optical illusions. If you have watched Brain, there was this show on Discovery. I don't remember the name, but they used to show these kind of optical illusions. So our eyes have this bias in it, that and it tries to see things with certain context. So now, okay, so you might ask, why are these optical illusions relevant for image analysis in life sciences? Look at these images here. You can see. Uh, what would you say if I asked you to compare the intensities again? Anyone? Nucleus A and B. Anyone? Okay. I have another question. Which nucleus is larger, you think?
It looks like A is larger. So
Yes. Yes. But
both are
uh, yes. So it will appear to you like that. Uh, but the nuclei A and B are equally bright, and the size of the nuclei A and the cell A and B is also same. So this was done after doing quantitative analysis of the image through mathematical operations, and not by just looking. So our eyes can be deceptive at times. That's the whole point I am trying to say. So we cannot fully trust our eye. Therefore it is always better to use unbiased image processing routines in analysis tools. Okay, so now I'll talk about what is a digital image. After that brief introduction of visual perception, how biased it can be, as well as the benefits of quantitative image analysis, we will dig a bit more into the properties of digital images. What is a digital image, and what is the main difference between a digital image and an analog one? Okay, now the definition of digital images. Digital images are composed of small addressable units called pixels. You can see here. Typically, these units are assembled in a rectangular manner, and every pixel is defined by its coordinates and value, which is called pixel value or pixel intensity. You can see here, these U and V are the coordinates, and this I is the intensity, and they belong to two different sets. And I cannot believe that I am talking maths here. Okay. Okay. Now, it is important to remember that typically only whole numbers are allowed. For the display of the images, we need to assign a color to an intensity value. For monochrome black and white images, it is common to display the lowest value, typically 0, and the highest value white. So we have a convention that I will talk about it later. So, but please keep in mind that this is just a convention. It is very tempting to think of pixel as a little square, but this is not entirely correct. It is more appropriate to see them as infinitesimally small sample units without a shape or a size. Okay, analog to digital images. Uh, you can see the transition here. Transition of an analog image to a digital one. How this image is stored. An analog image is consisting of a continuous value, which need to transfer into values with concrete steps in the case of a digital image. This is the case for the intensity, but it is also true for the distance and size of the pixel. I will also talk about these things in detail much later, but just try to understand what is the difference between an analog. The analog image is a continuous image. When we convert an analog image into a digital image, we basically uh, we break them into small boxes, and each box is assigned some number, and that is a convention that depends on us what we are doing. So I will also those kind of conventions I will talk about later. Okay. Okay. So it is okay. So see here that it is important to memorize that digital images are rastered images, being nothing else than matrix of integers. So what a digital image is, you can in a nutshell say that it is a matrix of integers. I will show you a few things to clarify my statement. Okay, now it is important to keep in mind that images and objects are distinct from each other. The aim of image analysis is to first analyze the image and to use the information gathered to come to a conclusion about the object itself. We need to consider that the image of the object is only a representation of the object. Its appearance and properties are highly depending on the imaging system. It is extremely important to know the properties, or if you formulate it a bit more scientifically, the transfer function of the imaging system. In every lens-based system, the image of an object is the convolution of the light emitted of the object and its surrounding, with the response function of the imaging system. How important the transfer function is illustrated by the two images shown here. The same object, so-called diatom, was imaged with same microscope 2 times by varying the aperture stop of the microscope. The result, uh, two completely different images of the same object. This example uh, illustrate two facts. First one is an image is always only one out of many representation of the object. And the second one is it is important to know the imaging system as well as its settings. Since by changing the aperture, we can change how the representation of that object is looking. So that's the point. Now, uh, I will talk about a bit of physics here. We have studied these things in our first year as well. Microscopy is often called diffraction limited. The knowledge of this response function is necessary in order to interpret the obtained image correctly and meaningfully. In ideal case, it can be retrieved from the metadata of the image, which means in essence that relevant information of the acquisition is stored along with the image. So basically, when we do microscopy, there are certain, I will talk about image formats later. There are certain image formats which allows us to store some data along with the image, which is called metadata. And the metadata contains all the information about the setting of the microscope that we used while doing this microscopy. So that when we do it again, we will have information that under what condition and what settings we took those pictures. So it is also important in scientific world to tell people that what information, what settings you were using while doing the microscopy. Okay, so here I'm showing here two diffraction limited spots, and its distance is decreased from left to right in the upper as well as in the lower row. One may debate when the 2 points are not distinguishable, but it is obvious that it is impossible to identify two objects in the lower row, right? Here the resolution of the system is not sufficient. The bigger the spot size or less resolution is expected when we decrease the nuclear aperture of the imaging system or use longer wavelength. So you might have studied this formulae, right? Diffraction limit, relays criterion for uh, uh, image resolution and things like that. We have studied those in our first year as well. Now, the second important thing is digital sampling. What does that mean exactly? It is uh, it is the emulated image which serves to explain the resolution. Now we have added one layer of complexity and changed the sampling frequency. So sampling frequency is also known as pixel size. So you can change a setting, like you can show 5 nanometer per pixel, you can show 8 nanometer of distance per pixel. So you can change the settings to show show the distance by each pixel, and that would affect your image. So not too surprisingly, we are losing resolution if we increase the pixel size. You can see here we are increasing the pixel size and we are losing the resolution. Uh, in particular, the image in the right corner is illustrating it more finely. So you can see that we cannot distinguish two different dots any longer after increasing the digital sampling size or the pixel size. So it is also important to keep in mind what pixel size you are using. Those who do gaming and all, they know these kind of things. You change these pixel size to reduce the size of an image. But if you go beyond certain level, the image will become useless because it won't have the relevant information that you need for analysis. So you can see how this like with increasing with decreasing the increasing this pixel per per length per pixel, the it is becoming even more difficult to distinguish between two spots. Now, these were some of the important concepts that we needed to understand this talk. Now I will go to image acquisition workflow. So now we have discussed the individual steps of image acquisition workflow. So let's assemble it. An object is imaged. The produced image is detected. You can see here. This results in a raster or digitally representation of the image of the object. You can see how we have a continuous like, image kind of thing, then we rasterize it and convert it into a um, raster digital image. So this is the hallmark of a digital image. The response of the imaging system as well as the parameters creating the rasters are important. We have seen this, that is resolving power and the pixel size. The functions involved to display the images are also extremely important. They will be covered later. So it is not just acquisition functions are important. How we uh, display those function or data is also important. So we have certain functions that we can build our on our own to display image. We have a different set of function to create a digital image, and then we have a different set of function to display it. So this step is controlled by that. And we can modulate this function according to our convenience, and we will see later how we can do that. Okay, so this is done. Okay, now I'll talk about some image file formats, which is also important. Okay, I forgot to mention here one thing. Each step can be considered a mathematical operation, and in order to interpret the resulting images correctly, one needs to know the operation as good as possible. This is why it is important to know the properties of imaging system, including its detection devices and ideally stored as metadata with the image. So this is something I have talked already. Okay, now at some point we will have to save the data, right? Which we have acquired digitally. And this poses the question, which format shall be used? What are the advantages and pitfalls of the different imaging formats? The most common format is JPEG. We have heard about it many times. It is extremely versatile for non-scientific images uh, uh, because it is an extremely clever compression algorithm. So basically, we use a mathematical algorithm here to compress the image, and in order to do so, we lose some data. But those data we cannot see through our eyes. So I will talk about it. The most common format, JPG, is shown here. Uh, so basically, the data is lost in case of JPG because we compress it and we throw some data in order to compress the data. And that's why it is called a lossy compression. You can surely imagine that such data loss is unfortunate for scientific data processing, right? Therefore, using this format is not recommended for original data. However, for presentation and reports, the conversion can be useful. We generally use here both these, like the entire thing is a JPG image and it is compressed. So it doesn't matter. I'm just showing here for representation. As the compression is not visible for the human eye. Like we cannot see this compression. The effect of compression is not visible to a human eye. So that's why we use JPEG for representation generally, because the data that we have thrown from this image is not detectable anyway through our eye. Okay, now the most preferred file format for images in science is TIFF, which stands for tagged image file format. It is a lossless compression method, so the original data can always be restored. It allows to link a tag in the image, and these tags are typically used to encode the metadata. I have talked about it. Like metadata contains information in what condition you have taken this image, and this file format allows you to store that kind of information. So that's why most of the scientific community use this kind of file format. Now, so now I told you that earlier that uh, images are created and then it is displayed by two different functions, and it is the display function which makes uh, digital analysis of image very convenient. So uh, now we know that from a real object to digital image, we have to go through an imaging system and a detection system that does the rasterization, which means the discretization of the real object into the digital image. This classically give rise to an array of value at different coordinates. We have seen also that, which can then be displayed on our screen. This means that if we take a look at the underlying data that represents the image, we should see nothing but numbers. So really an image in this case is nothing more than a 2D array of numbers which where each cell is what we call pixel. Okay, so now I will show you a demonstration. Okay, So new. So this is image software. It is also called Fiji. I will create one image here. I will draw something to show how the inside of the image looks. Let's say I have drawn this, something like this. Now I will save this into a text as a text file. Okay, desktop. I can save it on desktop. Save. Now, when I will open this thing, you can see that this is nothing but a matrix of zeros, and wherever I have drawn those things, you can see there is some value. So this is nothing but the pixel value for that pixel that I have like drawn. Like when we draw things, we basically change the pixel values. So I have told you before that there is this convention that black is assigned 0, and in an 8-bit depth image, uh, pure white is assigned to 55. So this is a convention that we follow. And the in between gray values are lies between 0 to 255. So this is one example that I was going to show you. Now coming back to the presentation. So this is, so we have looked at a digital image is just a matrix of numbers. Okay. Now, so we have 2D arrays of pixel and we know how to display it. Now the interesting thing would be to be able to get some statistical information out of it. We have a matrix, but we would like to extract some statistical data from that matrix, and that is the entire point of this talk. Like if we can extract some information from that matrix and use it for some analysis, then it would be really helpful for us. So now the interesting thing, okay, now what else? Yeah, because digital images aren't just about show and tell. They contain enormous amount of information. Okay, this is done. At first, a typical approach would be to plot the distribution of the pixel intensities. So we have plotted the pixel intensities in the hologram. Here you can see the 0 corresponds to the darkest, like the blackest value, and 255 corresponds to the whitest value, and in between values give you the gray regions. So, and here we have plotted histogram, like how many, like let's say we have some kind of gray value here. Let's say we can call it 100. So how many pixels correspond to this pixel intensity? So this is the distribution of pixel intensity in the form of a histogram. The histogram does not provide any special information and on where the pixels are, but just how many pixels of a certain value on the image. On top of that, you can see that you can get a lot of information at a glance from the histogram, such as the mean intensity. You can see mean intensity here, the standard deviation count, min and max values, and mode count. Like there are several information that you can see here, which can give you certain initial information. Now, it is interesting to note that if we focus on the first bin, this one, the first bin of the histogram, we seem to have a lot of pixels that are exactly 0, while not unusual in itself. It is odd that distribution is not continuously growing from zero. So this is a red flag. If things are not continuously growing, then there is a red flag here. And I will tell you why. There seems to be a gap in the histogram. So there is a gap here. And this is because this image presents an artifact in the bottom right corner here. We have an artifact. So where there is, this is a black strip, right? This artifact is a black strip, which is it is most likely not part of the image, but rather linked to a problem during the digitalization of the image. So it is inevitable that during all those acquisition process and digitalization process, some error might happen. So by looking at this histogram, we can detect it. So, which lead us to an interesting conclusion that we can discover artifacts with the histogram by looking at the saturation at the ends. So basically, if you have any kind of saturation, let's say black saturation or white kind of saturation due to image detection error or digitalization error, by looking at the 0 bin and 255 bin, you can tell if, sorry, excuse me, you can tell whether you have saturation in your image or not. And there is a guideline that how much saturation is permissible in a scientifically acquired digital image. So you have to follow those guidelines or your image won't be considered legitimate. Now, I'll come to seeing colors now. The retina is the part of the eye which contains the photoreceptor cells. We all know that. The image formed by the lens is projected on this layer of cells. The retina of vertebrate has two types of photoreceptors. The so-called rod cells are mainly responsible for situations where we have to deal with low light levels, and so these cells have much higher sensitivity than the cone cells that deliver black and white images. So rod cells are more sensitive toward black and white images. And now what about the colors? For seeing colors, other photoreceptors are needed, the so-called cone cells. These cells exist in three spectrally different variants. The spectrum of the rod cells covers the range from 400 to 800 nanometer, which is therefore also known as the visible range. The relative sensitivity of the three different cones cell is plotted versus the wavelength. Our ability to see colors from the fact that a given wavelength is stimulating each cell type to a certain extent. Basically, we see different kinds of color because all these three different kind of photoreceptors are stimulated at different levels. So let's say blue is blue is uh, blue is stimulated at an intensity of power 10. G is at power 20, R is power 30. The combination of these three will create a unique combination of color. So basically, this is how we see things. Cone cells are stimulated. Each type of cone cells are stimulated at different level, and that creates different kinds of combination of um, sensation of this color. Okay, so this is the concept that has been copied to construct one of the most popular color system to represent colors. I think you have heard of. I think you have heard about this, right? RGB color space. So basically, we use a coordinate system kind of thing. At one axis, we have blue from 0 to 1, like this is a scale. On another axis, we have red, and on the third axis, we have green. So like we have like we have a coordinate with 3 values, and each coordinate will have a unique color. So let's say here we have at this vertex we have the maximum blue stimulation and maximum red stimulation. You can see in this case the color is magenta. In case of where there is no all the three photoreceptors, or like not photoreceptors, like all the three scales are 0, we see black color, and here when all the three scales are 1, we see white color. So basically this is a coordinate kind system kind of thing, which was developed to see colored digital images. Okay, so I told you that we follow these conventions, and we follow these conventions, and each convention is is made by certain mapping functions. So we have certain mapping functions with their own convention. Like you can see, you can assign black to 0, or you can assign pure white to 0. You can assign a gradation of red to this scale, and so on and so forth. And by doing so, you can display the same digital image in a different way. I will show you a, a, a thing, a demonstration to clear it a bit. So in order to avoid modifying data, the concept of lookup table, so LUT stands for lookup table, has been introduced. LUT is a mapping function which allows you to display a colored image without the need to modify the underlying original data of the image. So I will just show you something that would help. So I have already written some codes so that I can run it and show you some results beforehand, so that it will save some time. So here it is, LUT. Okay. Okay, so these are different mapping functions, basically different LUTs, which are nothing but different kinds of mapping functions or conventions in which you can show these digital images. Now, here we have a digital image, which is called blobs. I will show you how this looks in different kinds of display functions. So here, this is a display function called fire. This is grays. This is ice. This is spectrum. This is something 332 RGB. So you have different kinds of display function available, which follow different conventions. And this is how you represent digital images. Basically, you don't change the underlying data. You just change the function which maps it to the screen. So we have studied these kind of mapping functions in our M101, right? So this is just similar thing. So I will close this and I will show you more demonstration. Okay, so here we are. Okay, so now I will just talk to you through demonstrations. I have introduced you to basics of image processing and analysis. So these are some of the important concepts that you need to know before doing image acquisition or analysis or extracting information from it. You need to know all these theory. So I will show you some image math now. Okay, so, that I would show you through demonstrations. So basically, so we have seen that digital images are nothing but matrix of numbers, and we can use those matrices to 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 say something about two different images. Let's say we have two different images from two different time points, like two different images of the same object of the two different time points. Let's say a protein is accumulating inside a cell, and we want to know at what rate this protein is accumulating. So what we can do, we can take image of one time point, and we can which is nothing but a matrix of number, and we can subtract this matrix from another to from an we can subtract this matrix from one matrix to another matrix. So basically the difference between the two matrix, which is nothing but digital image of two different time points, we can get the information that what is the change of intensity. And this is just one of the example. You can do ratios, you can do you can in fact different kinds of filters that we see on Instagram or any image processing softwares, those are nothing but 3 by 3 matrix or 4 by 4 matrix, 5 by 5 matrix, and they multiply. So when we apply a filter, we multiply those filter matrices to the original image, and by doing so we transform the image. So we have studied this transformation process.
Shubham, excuse me. Are
Yes.
you showing any images right now?
I'm not showing, but I'm just talking. I will show.
Okay, okay, that's fine, that's fine.
Okay, so I have some codes here that I have written. So I will show you an example of ratioing to image. Okay, so please open ratio demo image. Okay, ratio demo image. Yes. So here you can see we have an image, and it has, sorry for the noise, the Bhavali is here. So it has time points here, and we are trying to see, we want to calculate the ratio of the intensities in these cells. So we have two different images, and that means we have two different matrices, and these two different matrices correspond to two different time points. And now we want to compare these two matrices by calculating the ratio. So let's get going. So you won't see the code. The code is hidden inside the background. The code has all the mathematical operations. I can show you the code later, or I can send you some as well. So here you can see that that I have calculated the ratio of the image, and wait a second, tile. Wait a second. Yes. So what we have done, we have just divided one matrix to another, and see, uh, you can see when I put my cursor here on this cell, the value of x is 42 and the value of y is 85. It is going to be seen away, like you will see this here, and the ratio will be also be present here. So when I will move my cursor, you can see that I have created a pictorial demonstration of the how like the how protein accumulation is changing in, let's say, two different cells. So like these kind of visual depiction gives us a lot of power. Like this is just one example. uh, you can think of many such experiments where you can use this technique to get a visual depiction of what is happening. So we have created a scale here. So it is nothing but a mapping function. So the ratio value is if the ratio value is decreasing, it is going to more towards black, and if the ratio value is increasing, it is going more towards yellow. So you can have a like some kind of sense with these kind of operations, and these are all mathematical in nature. Okay, so I will show you some more to give you some more idea. Those all. Okay. Plugins. So I have, so this was my work in the summer. I have created many such programs to normalize stuff. I can show you another thing first. Image process, image calculator. Okay, so for that I need to do some work. So I'm going to show you how subtracting two image can help us. Like can help us in a way that that is going to help us. Okay, so this is one of the example. Like this is one of the image, and I will open the another one. Pen. Yes. So what are these images? So basically, we have image. This is a bright field microscopy. We have imaged a diatom here. Diatom, you must be knowing about. It is an organism, single cell organism. So in first image, you can see the diatom. In the second image, you can see there is no diatom. So we have just imaged the background, like without the diatom and with the diatom. Generally, we tend to have background noises, and that interferes with our information that we want to extract from the image. So what do you think I'm going to do here? Can anyone say? What do you think I'm going to do here? I have one image. Yes, hello.
Subtract
Yeah,
the background noise to
delete
Yes,
the
get
background.
accurate.
yes. So
Delete the background.
basically we have two different images which are nothing but two different matrices, and we don't want the background matrix to interfere with the image matrix that we have, because we want to extract information only from the organism that we are looking at and not from the background noise. So we can just subtract these two images and we'll get the image will become noise free. So I will show you how this works. Image calculator, image one, yes, and image two is this. And what is the operation? We are creating difference. Create a new window. And okay. So can you see how amazing it looks, right? So this is everything but our like background noise. So you can understand the power of these mathematical operation, and you can yes, hello,
Hello,
yes.
Shubham. Yes. So, how are you able to separate uh, the background noise with the actual picture, or were these two pictures taken separately?
Yes, these pictures were taken.
So,
These
what?
pictures were taken separately. One like this, like let's say we have a mounting agent. We generally mount things on a slide, glass slide, right? So we put a mounting agent on glass slide. Then we put the organism through a pipette. So what we can do, we can prepare two different slides, one without the organism and one with the organism, and we will use the same solution. We are not going to use two different solutions. So so that would help us to there might be some fluctuations that might be there, but those are going to be like very minimum. So are you getting my point? Like this is the control and the experiment kind of thing. We always have a control, right? Negative control or positive control. So that's that. Do you understand it?
Okay, but given an image, can you separate the background noise? You have just one image, and then there's a background. How would you separate the background from what uh, from the actual picture that you really want,
Can you repeat
whatever
again?
experiment?
Can you repeat again?
Uh, okay, sorry. So you have a picture. You have just one picture with you. The picture has background noise. Now, with with just that one picture, how would you eliminate the background noise? Uh, do you
Okay,
know
so
about this?
yes, yeah, there are algorithms for that as well, but that involves Fourier transform, and I will come to that. Uh, there are methods to do that.
Okay,
So
go ahead.
basic,
Thanks.
okay, so like those are very complicated mathematical operations, and like, but they are like very powerful tools as well. Okay, hello, yes,
Hi.
yeah,
Hi. We can do, like one can do background subtraction. There is an option in processing. So in background subtraction, one can do subtract the background.
okay. Oh, I don't know, maybe like from just one image.
Yeah, from one image. So there are two options. One is math and subtract. You
Okay,
uh, just look at that. One can do that also.
okay, math. Okay, so the speaker who is speaking right now is the PhD who has taught me. So yes, she has. Yes, this you are talking, right, or what? Hello,
Yeah.
or what? And in background. Okay, so I'm not aware of many of the functions here. I'm still learning. I have learned this one only as of now, but there are many functions here. You can see how, what, like this is fast Fourier transform. This is filter. So there are many ways like you can ch- change these matrices, and I am still not aware of like many of these functions, and I'm still learning, but I have given you an idea that there are books available on in this entire software. So by reading those books, you can get an uh, idea of how to use this. Okay, now I will show you some more examples because the time is running out. Uh, run. I will show you some more examples. Okay, so I have done ratioing. Normal stack is one another thing. Gaussian, should I do? Okay, so let's say. Okay, I will show you Fourier transform now. How Fourier transform works, and this is one of my favorite example. Like I I studied about Fourier transform uh, like when like because of one of my friend, like he inspired me to study Fourier transform. I forgot to if Gautameshwar is present here. He was the guy who inspired me to look into this thing. So what was the so Fourier transform is very important, like very interesting tool that is like if you know this how to access this, you can basically do magical stuff in this software. So I will show you how. So here we have a electron microscopy. Oh, sorry. Electron microscopy image. Wait a second. Wait a second. So here we have an electron. What happened? Yeah. So here we have an electron microscopy image, and this image was taken. This image was not taken well. Like it was taken not in a well maintained microscopic environment, or the acquisition functions had some error because of which you can see there are some lines, and this is not due to uh, this is not due to uh, some experimental error. This is due to like fluctuation in the environment or the illumination or the acquisition function. So like these are so so basically you can see that there is some directionality in this. I'm trying to come to Fourier transform here. There is some directionality in this image, and we can use this math. We have some mathematical operations that can calculate the directionality. Like the image in general does not have that much directionality as compared to the noise. The noise here has some directionality because it has these parallel lines. So if we can, devise a mathematical operation which can, calculate. Uh, so I need to give you some insight of Fourier transform first. So basically, what Fourier series or Fourier transform? Uh, Hello, Bhaiya. Would you like
Yeah.
to explain Fourier series and Fourier transform a bit?
No, no, I would not want to waste anybody's time here, but you go ahead. Thanks.
Okay, so basically every frequency in the nature can be represented uh, by a sine or cosine function. So this image has multiple signals in it, and each one of those signals can be represented by a sine or cosine function. And by adding those different sine and cosine function, we arrive at this image. Now, what we need is to identify the sine or cosine function corresponding to this, uh, noise. So, and by identifying those sine and cosine function and subtracting it from the original image, we can remove this uh, noise. So here it is. So you can see that this is the image, and a second, what just happened? Yes. So this was our image, and this is the Fourier domain. So Fourier domain is an entire space of its own. We go from time domain to Fourier domain. Fourier domain is this image gives the visual representation of Fourier domain. So I have this code in the background which calculate the directionality in the Fourier domain of that those frequencies that which correspond to these line signals. So I have not written these codes. I have acquired it from somewhere because I am not well versed in Fourier transform. Maybe I'll take some course next year to understand it more. But I'm still learning, and uh, so basically that the code just identified what is the contribution of these lines in the Fourier domain, and this yellow line depicts the contribution of this disturbance signal. Now, when I will create the angle was determined to be this, we will now create the Fourier mask. So now I'm trying to create the Fourier mask here. This is the Fourier mask, and we will subtract this Fourier mask from the Fourier image of the original time domain image. And yes, so basically what we did here is, so you can see after doing this Fourier analysis and subtracting those noise signals, we have got something very interesting, right? So what happened here basically? What we did here is we went from time domain, like we had this original image with some stripes. We went from time domain to Fourier domain. In Fourier domain, we tried to identify those frequencies corresponding to those directional signals, and we identified it. Then we created a mask corresponding to those regions, and then we subtracted that mask from this Fourier image, and then we did inverse Fourier transform. And when we did inverse Fourier transform, we got this image. So any questions regarding this? Did you understand the workflow here? Hello?
Yeah, I think we all understood it.
Okay, so what I want to say is there are many mathematical operations. I'm still not aware about many, and I'm still learning about many of those. And by knowing some of those, you can have enormous power over your digital image, and those can help in your research, especially if you deal with image images, digital images. So uh, so so basically now I will talk about segmentation is nothing but identifying your object of interest from your. So let's say we have an image, we want to uh, we want to let's say we have an image. Saw earlier that we had an image of cells, and we need to identify those small circles from the background. So segmentation techniques help us in order to do so. I will also show you some codes, how we identify our object of interest from several other objects that is present in the background. So detect cells. So this is one of the code through which you can detect cells, and which is nothing but how you can okay, there are no images. I have to open the image first. Speeds. Let's say this one. No, this is not good. I'm hoping some HeLa cell images, and I will show you how you can. So here we have an image, and it is present in different time points, but we are going to do it for the first time point only. So we want to we want to separate these cells from the background, because by separating this we can calculate the area of these cells, we can calculate the perimeter of these cells, and we can. So how we can do it mathematically? Like we won't like to do it manually. That would be really tedious work. So there are ways you can do it in a mathematically, and that would be very easy. By writing some simple code, you can do. So this is one of the code that I have written, detect cells. So the code just ran. Now I will show you some of this. So you can see here the code ran. It detected different cell region, identified those regions, and now we have it is showing some results as well here. So we can see here there are 23 counts of cell in this image. The total area of the cells in the image is this, and the average size of the cell is this. Percentage of area that the cell is occupying is this. The mean and the mode. This is showing you what? Mean and mode. This is showing you the pixel intensity. Ignore this. So basically what I'm trying to say, there are, like, this is just one of the way you can calculate perimeter, you can calculate standard deviation, you can do a lot of different kinds of calculation, like process. I can show you what analyze, right? Analyze particles. Okay, like I am going to show you what else, what parameters can you set measurements. Yes, this was this. Yes, so you can get in result these kind of things. Right now, I had selected area, standard deviation, min max gray value, area of fraction, model gray value, main gray value, center of mass, perimeter, centroid, skewness, kurtosis, median. You can do all kinds of calculation in this software, uh, and this is all done using codes, and this makes our life very easy because we just need to write a code for one image, and now we will apply that code to an entire batch of images, and that would save us a lot of time, because if we'll do it manually, it will be a very you know time taking. So that's the whole point of doing all these things. uh, so uh, okay, that's it. I also told that I will talk about artificial intelligence and deep learning algorithms. So, uh, so I will. So basically, what is an deep learning algorithm? In normal coding, we write algorithms by ourselves. But we have a certain set of operations and in our mind, and we write our codes according to that. But in deep learning algorithm, the coding part is done by the computer. We just need to feed in some input image and output image. When we feed, so we give computer some input image, that this is the input image, and then we have some like which is called test image, and then we have these two was like we have, sorry, test images. Basically we train the artificial intelligence to find the area of interest for ourselves, and by artificial intelligence, basically they write code according to input and output image. So we feed in some input image and we also feed in some output image. The artificial intelligence compare these two images and write codes according to this, and create a function that would lead us from input image to the output image. And so this is how deep learning algorithms are revolutionizing this process, because we no longer need to write the code uh, any anymore. But this does not mean that it does not have limitations. Each of these programs have certain level of robustness. So artificial intelligence give you some program by comparing input and output image, but those programs may have certain level of robustness that you need to check by feeding in different images, input and output images. And when you reach certain threshold of standard robustness, then you say, yes, now I can use this software, sorry, I can use this code that has been provided to me by the artificial intelligence. So that's it. In acknowledgement, I would thank um, Professor Ranjit, who inspired me to work on this project. PhDs in his lab, especially Rosalind Didi, who has helped me a lot since my first year. Some of my friends who keep inspiring me, to go into multidisciplinary areas and learn things beyond my, beyond my capabilities, or my, what should we say? Okay, whatever, beyond my comfort zone. Yes, and especially Gautameshwar and some of my maths department friends who keep motivating, even though I was deeply motivated in first year due to maths. But anyway, my elder brother, who keeps scolding me all the time to do stuff, and I think that's it. And some of the books that I have read, and some of the online courses that I have attended. So yeah, that's it. So if you have any questions, you can ask me now.
Thank you, Shubham, for that presentation. That was really well presented, and I personally learned a lot. Okay, so does anybody have any questions?
Hello, Shubham.
Yes.
Yeah, in that Fourier transform part, so you have an
Yes.
image. You take the
Yes.
Fourier transform of the image, and
Yes.
from the Fourier image, how are you separating the noise?
Okay. So what there is I have this code which calculates the frequency corresponding to some directional noise in the background. So since you are aware about the Fourier transform, right? There are different kinds of frequencies associated with each sine and cosine function, right? Hello.
Yeah,
Uh,
yeah.
so what we saw in that image, that there were lines. Those lines were the noise, and those lines had some directionality. They were lines, right? Parallel lines. Are you
Yeah,
are you getting that?
yeah. I'm
So
getting. You continue.
so I wrote, like, I didn't wrote, someone wrote a code to find frequencies corresponding to those directional lines. I will send you the code. Maybe you can understand it better since you have studied Fourier series and Fourier transform much better
So,
than me.
but if our image, the thing we want to observe,
Yes.
that also has some directionality, then that will also get eliminated, right?
Yes. 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 So
So
you
we
will.
have to customize the code.
Yes. So
Okay,
so
okay.
there are yeah, there are certain limitation to this method as well, but sometimes, like, you don't need the code is only changing the intensity of the pixel, and we are not analyzing the intensity here. We are just looking at the, electron microscope. We are just looking at the structure of the surface. So it doesn't matter because we are not looking at the intensities. It will diminish the intensity, of course, but since we are not looking at the intensity in different regions, we can go, like, it depends on the situation. What in different situation you can use different mathematical operations. So does that answer your question?
Yeah, it's fine.
Any more question?
I think that brings us to the end of this session. Or does anybody have any questions? If not, then we can stop.
Okay.
Thank you, Shubham, for the wonderful presentation. I had the next presenter, whoever it is, will have a very high bar to cross now. So, with that, we end. So, bye, guys. I hope you have a great weekend and